In part 6 of our notes, we are now moving on to learning target 3. Students will be able to demonstrate the process of translation by using the genetic code to match messenger RNA codons with amino acids. So we're going to be really focusing on the process of translation, identifying characteristics of the genetic code, but also being able to demonstrate how to use the genetic code. All right? So we want to start out first by just talking about characteristics of the genetic code. Some things about the genetic code is that it's a triplet code. That means every 3 bases on a messenger RNA, or every 3 nucleotides, codes for a specific amino acid. Okay? And remember, the amino acids are the subunits or monomers of a protein. So now that messenger RNA has that code, it can use that to help kind of signal what amino acids need to be put together to make the protein. It's a non-overlapping code, so when we're looking at messenger RNA, we read it, the reading frame we mean is every 3 bases on, or nucleotides on messenger RNA, that will code for one amino acid. The next 3 will code for another amino acid, the next 3, and so on. So it doesn't overlap. It can specify things other than amino acids. We do have what are called the start and stop codons that kind of signal the beginning of translation and the end of translation. This big term, we cover a little bit more in genetics, so it's not so much that you need to know it, but it will be a term that will come up later if you are to take genetics or maybe AP Bio. All it means is the genetic code being degenerate means that more than one codon can specify the same amino acid. And it kind of gets back to it being a triplet code, and I'll talk about that in a minute. But a really cool thing about the genetic code is that it is universal. That means every living thing that they've discovered so far has the same genetic code. We use those same nitrogen bases. We have the same amino acids. Now, how different organisms put together those amino acids to make different proteins is different, but we have the same genetic code. And that really is important because it allows us to do a genetic engineering where we can take the DNA from one organism that codes for a trait and insert it into another organism. So kind of interesting, and there's a lot of applications with it because it's universal. So what do we mean by the genetic code? If you've looked at in the poster in my room before, and those of you who are virtual, you of course can't see that, but this is the genetic code. Either this box, this is kind of old school, this is what I grew up with back in the day when I took biology, many years ago. About what? 35, about 37 years ago. That's kind of scary. But here's a new one they came up with right here. But basically, it shows you the different codons and the different amino acids that they code for. So all these big fancy words that are out here, those are the different amino acids. And there's 20 different amino acids that living things have. And the reason we have a triplet code is if we would have only had a single code, we only have 4 bases to choose from, A, U, C, and G. Again, we're dealing with the RNA bases. That would have only given us 4 codons, which means only 4 amino acids possible. We have 20 here. We need 20. If we had a, oops, I forgot to change this each time. This should say double code. And this should say triplet code. All right, sorry, I goofed on that. But if it's a double code, now we take 4 times 4, or we square it. Now you can come up with 16 different codons. Well, that would only give us 16 amino acids. How many do we have to get again? We have to get to 20, don't we? So the reason we have a triplet code for our DNA is if you cube the 4, or if you take 4 times 4 times 4, then you get 64 possible codons. Now we have more than enough to cover these 20 amino acids. And that's why, remember that big word degenerate, that you have several amino acids that have many codons for them? Let's look here. Look at proline here. Look at CCU, CCC, CCA, CCG. So a lot of them will have multiple codons that code for the same amino acid. All right? And we have some that signal those other things like stop codons. Methionine is what we call our start codon. All right? So a lot of characteristics of the genetic code. So how do we use it? I like using this wheel. The genius who came up with this is so much easier than using that rectangular box. But you always start in the center. Remember, you always have a triplet code. So let's say our codon is AUG. Well, we always start in this central circle first. So here's my A. Then I go out to the next ring. I'm only going to stay in this quadrant because that's where the A is. U is the next one. That means I stay in this piece of the pie. Here's your U. Now I see it's a G. Here's my G right here. And that means methionine is what AUG codes for. So let's look up GCA. Now we're right here in this quadrant, so we can only stay in this area. C is the next one. That means we go to this piece of the pie. Here's our C. And we see that all of them won't even matter, but A, GCA, codes for alanine, and there's your answer there. So you need to learn how to use this genetic code wheel because a lot of our worksheets coming up are going to have you do some of that. And your quizzes and your test will also, you will have to know how to use this genetic code wheel. Okay, so again, some codons are stop codons. I showed you that earlier, that signal the end of translation. And some of them have several codons. So like leucine, for example, LEU. Look at that. We have 4 right here, and we have a couple more right there. So 6 different codons code for leucine. So the actual process of translation, the visual you should look at here is that RNA is going to a protein, meaning messenger RNA copy is used to assemble amino acids into a protein. That's what translation is. We're using now that copied message on the messenger RNA to put together amino acids, which are the subunits of protein, to make that protein. This occurs in the ribosome. That's where proteins are made, remember? So now we're on a ribosome. Every 3 nucleotides on the messenger RNA specifies a particular amino acid. So we call that a codon. A codon is a series of 3 nucleotides on messenger RNA that codes for a protein. Okay? Or that codes for a, boy, I'm making all sorts of mistakes today. Codes for an amino acid. Okay? An anticodon, here's another term, anti means the opposite. Anticodon is a series of 3 nucleotides on transfer RNA that's going to be complementary to the messenger RNA. In other words, it's going to be exactly the opposite of. That's why it's called an anticodon, because it's going to be the opposite of a codon. And it's kind of the job of ribosomal RNA to kind of help make sure those get paired up, to make sure the messenger RNA gets hooked onto the ribosome, and that the transfer RNA can bring its and match up its anticodon with the codon. Okay, so here's your messenger RNA. If it has a codon of UCG, using the base pair rules, the anticodon would be AGC. It has to be just the exact opposite. Okay. So as far as the steps of translation, the first thing that happens is the messenger RNA connects to the small ribosomal subunit of the ribosome. So here we have your messenger RNA here. Here's your small little ribosomal subunit here that's going to get connected to it. Then a transfer RNA brings an amino acid to the messenger RNA on the ribosome. So here you see that transfer RNA bringing that amino acid here. And remember, the start codon, AUG, is going to match up with the anticodon, UAC. And this transfer RNA here is bringing that methionine, which usually signals the start of translation. So here we go. We have The transfer RNA lined up, it brought its amino acid here, it matched up its anticodon with the codon. Here's the anticodon. And here's the codon. Okay? Then, for step 3, another transfer RNA will bind to the messenger RNA. So we have another one coming in that will bind to the messenger RNA right next to the original one. Once you get 2 of these right next to each other, a bond called a peptide bond forms between those 2 adjacent amino acids. So here we had the one transfer RNA that brought in methionine, which is abbreviated by MET. Then we had the next transfer RNA coming in, matching up its anticodon with the codon here. It brought in the next amino acid called leucine. Once these 2 were lined up next to each other, that peptide bond formed. Okay? And you'll notice too that the large ribosomal subunit came and hooked on too. That's that large ribosomal subunit that allows space for 2 transfer RNAs at a time to come in, allowing that peptide bond to form between them. And then those transfer RNAs, the large and small subunit shifts on down the line to the next open spot here. So it would just kind of shift on over here so that another one could come in and bring in its correct spot and bond another one in there. This process is going to continue. The transfer RNA will continue bringing in amino acids until a stop codon is reached. And that's going to signal the end. You see that this large and small ribosomal subunit down here, it's kind of shifted down the line and now it has another space. So this one's going to bring its amino acid. And you can see it just keeps going, and you've got this long chain of amino acids forming here until we reach a stop codon. That's going to cause everything to get released. This polypeptide chain here that's forming is going to start to fold, and then the protein is made and gets released. Okay? And the protein folding, the shape of a protein determines its function. So that folding part is a very important process at the end of translation. And there's different structures that the proteins can have. If we're just talking about the original amino acid sequence, we call that the primary structure. If it starts to form pleats or helixes or coils because of the interactions between the adjacent amino acids, we call that the secondary structure. And then as it gets further apart, some of these different amino acids will respond to water molecules and start repelling and whatever, and they're folding even more. That's called the tertiary structure. And then not all proteins get to this point, but if a protein consists of more than one polypeptide, then it will have, like this one has 2 polypeptides there, that will form what's called the quaternary structure. So again, in order to make a protein, you have transcription, copying the code from DNA to RNA, translation, using the code on the RNA to make a protein. And together, those make up what we call gene expression. And then we have our central dogma down here. So make sure you know all those parts for when you get to your quiz or test on translation. So all of our different parts here that we have to label, you have to label the ribosome or rRNA. So here's your ribosome right here. This is pointing to an amino acid. This whole molecule here is your transfer RNA. Remember, this side of it is called it carries an amino acid on one side and it has an anticodon on the other. And messenger RNA, this would be the codon, because this is your messenger RNA right here. Okay? And then these 3, let's look in our chart here. What would CUC code for? CUC is leucine. GAC, aspartic acid. And AUC is isoleucine. So that's how you use the genetic code wheel.